The issue with HIV eradication and treatment is obviously that there is rebound upon interruption of therapy. And it's still somewhat obscure, but not as bad as Ray was indicating, as to where the virus is lurking. And there are a variety of reservoirs, but the principal reservoir is in resting memory T-cell populations. Now, there's been an ongoing debate about how HIV gets into these cells and becomes latent, but I think the best idea and the correct interpretation is something that Bob Silicano enunciated a number of years ago, which is that when effector cells are infected with HIV, that's a very permissive environment, but these cells are pre-programmed to enter a quiescent resting memory state. And if a cell happens to have been infected with HIV and the cell is transitioning into this memory cell phenotype, then the virus becomes latent. And so the latency issue is a cell biology issue as much as a virus issue. And I'm going to be describing what we know about these underlying mechanisms that take the virus in the cell from this active replication competent state to the resting memory state and latency. So we are able to model this ex vivo, and there are a variety of different models people work with, but our version is outlined in this slide, where we start with naive T cells, activate and polarize them, and then infect them with a reporter. And this reporter has a gizmo in it that allows us to purify infected cells. And then the magic in the system is taking these infected effector cells and forcing them into latency in a memory phenotype. And the key ingredients in this transition are treatment with a defined cocktail, including TGF-beta, IL-10, and IL-8. And this recapitulates the transition of the cells into this memory phenotype. What that looks like in practice is shown on these flow diagrams, where on the top panels you can see the GFP reporter, which is an indicator that the cells have been infected. And on the horizontal axis in the top panel is HIV Nef expression, which we use as a measure of HIV replication. And so in the expanding purified effector cells, you can see they're high GFP, high Nef. In the quiescent cells, there's residual GFP expression because it's a membrane-bound protein. It doesn't get degraded, but there's no Nef expression. And after reactivation, in this case by stimulating through the T-cell receptor, essentially the entire population gets reactivated. And you can see the cell cycle parameters in the bottom panels from the same samples, that the effector cells are in cycle. The quiescent cells are really out of cycle in a G0 state. And after reactivation, they begin to re-enter the cell cycle. Now, when we do this as an assay for latency and its reversal, we've been doing this for a number of years, and this is data accumulated over about a 5-year period from multiple different donors, multiple different polarization states, multiple different activators. You can see that the quiescent cells are very low levels of expression. If you activate through the T-cell receptor, you get nearly the entire population reactivated. And if you treat the cells with other activators, you get intermediate levels of reactivation, various characteristic levels of this. So an HDAC inhibitor, SAHA, gives an intermediate level. Excuse me, treating the cells with TNF-alpha, which activates NF-kappa B, gives a very poor reactivation level. Now we can take this model and analyze what's going on at the cellular level with RNA-seq in great detail. And one of the things that's very obvious is that it's at the RNA level, it's the replication state of the cell that's the dominant factor. It doesn't matter particularly which way the cells have been polarized, but quiescent cells look like quiescent cells, expanding cells look like expanding cells, activated cells are activated cells. And you can also see from the actual data, you get a lot of data out of RNA-seq, that there's an example here of the polarization phenotype for Th17 cells and with the expression of ROR gamma C. You can also see with HIV expression that, as expected, there's minimal transcription in the quiescent cells. In fact, that axis has been expanded 5-fold compared to the other scales to show that there actually is a little residual RNA transcripts, but it's really very, very quiescent. Now, if we look at what's going on in these quiescent cells, there is a program of gene expression that's been initiated, and the major pathways that are involved are pathways involving estrogen responses, MYC, and mTOR glycolysis IL-2. This is very characteristic. Now, this is comparing RNA-seq data from our model with the defined cocktail, a model that actually we developed about 10 years ago involving feeder cells, H80 cells, which give cells that aren't quite as quiescent. You can see there are more red bars. And a very similar model was actually the initiating model for activation and quiescing, a model from Vicente Planellas, where in that particular model there's more evidence for senescence rather than quiescence and more of an antiviral kind of response. But in general, the bulk of these populations, the bulk of these pathways leading to this quiescent state are the same and shared in these different data sets. One of the things that really jumped out at us and has come as quite a surprise, and we're investigating further, is that this transcription factor, KLF2, is a very major marker of initiating this program of quiescence. And it shows up in all the data sets, and it's extremely well expressed. But what came as a kind of a shock to us is that merely infecting cells with HIV, with lentiviruses, initiates expression of KLF2 and initiates this program of quiescence. And so what's going on is that there's a antiviral sensing mechanism in these expanding T cells, and infection with HIV begins to initiate this shutdown pathway. Now you can see in the bottom right-hand panel, this is looking at, I think it was 40 or so RNA-seq data sets that from our lab and a bunch of other labs, that this upregulation of KLF2 is seen almost invariably after HIV infection. You see it with some of the RNA viruses, none of the DNA viruses. And so it is something that is a sensing mechanism early involved in these lentiviral infections. So HIV is a co-conspirator in terms of getting the cells to begin to shut down and enter into latency. Now, at the viral level, latency is in essence a transcription problem. When the cells are resting and shut down, there's no HIV transcription. If you activate through the T cell receptor, you go from a state of the virus being completely off to being completely on. And the reason why it's so binary is that HIV encodes its own transcription elongation factor, the TAT protein. This initiates a positive feedback loop that activates transcription fully. So at the cellular level, what's going on is transcription initiation factors, Nfat, kappa B, PTFB, which is the cofactor for TAT, go down. Histone epigenetic silencing factors go up, and that leads to a viral state where there's no TAT expression, no HIV expression, and heterochromatic structures formed on the template. When you reactivate cells, you reverse this process. The transcription factor get reactivated, and TAT gets initiated, TAT production gets initiated, and the virus runs off. And so because of this autocatalytic mechanism, you never see intermediaries. It's either off or it's on. Now, in primary T cells, the dominant transcription factor is Nfat. Much of the literature about HIV latency is based around NF-kappa B, and that comes from studies working in cellular models, including Jurkat cells. I've got bad news for you. You have to forget about kappa B. It makes a small contribution to reversal of latency in primary cells, but the dominant factor is Nfat. And if you look at the bar graph in the lower right, panel C, you can see that if you treat cells with inhibitors of cyclosporin, you pretty much wipe out transcription reactivation. If you treat with an inhibitor of kappa B, then there's only a 50% reduction of transcription. So it's really Nfat that's driving the initiation and is the primary stimulus for transcription. The other thing that's important when we reactivate cells is the metabolism of PTFB, which is the essential cofactor for HIV TAT. And so in an activated cell, there's a pool of PTFB that's associated with an RMP complex called 7SK, and this delivers PTFB, TAT delivers PTFB to the promoter. In a quiescent cell, PTFB is disrupted, and I'm going to show you data showing that the catalytic moiety of PTFB, CDK9, is present in an inactive complex in the cytoplasm, as diagrammed here. So the cofactor for TAT, as well as the initiation factors for transcription, are reduced or gone in quiescent cells. And so reversing latency means reactivating these cofactors. And the additional thing that's going on in the quiescent cells, probably because nature abhors a vacuum, is that when the virus is no longer transcribing, then epigenetic silencing factors accumulate on the LTR and further block initiation. So here's the data that shows that PTFB is absent in resting memory cells. So in the top panels, you can see CDK9, this is the catalytic subunit of PTFB, and it's present in the cells, and it's in the cytoplasm. That's something you don't see in transformed cells, but is very characteristic of resting memory cells. After activation through the T cell receptor, then you get some enhancement of CDK9 production, and it accumulates in the nucleus. Cyclin T1, which is the other subunit of PTFB, is absent from resting cells. And activation of transcription involves resynthesis of cyclin T1, When cyclin T1 is resynthesized, it forms a heterocomp duplex with CDK9, assembles with 7SK, and goes into the nucleus. So here's some biochemical evidence for this kind of mechanism, that the CDK9 in the cytoplasm is associated with the chaperone proteins, HSP90, CDC37. It's in an inactive complex, and you can block this pathway of reactivation with inhibitors of HSP90. So this leads to when you activate a quiescent cell, you have to reactivate this pathway. Now, associated with reactivation of this pathway in PTFB are a number of phosphorylation events, and one of the ones that we've worked with pretty extensively is phosphorylation of CDK9 on serine 175. And we like this as a marker because this modification only takes place on PTFB that has dissociated from 7SK RNA and is transcriptionally functional. So this is the business form of PTFB. And you can see that from the micrographs that it's only present in the nucleus in a punctate pattern, and that in unstimulated cells, none of the PTFB is activated. In stimulated cells, all of the PTFB is activated. And it's an enormously strong correlation. We've got hundreds of flows with tens of thousands of cells, so billions of data points showing that unless you activate PTFB, you do not get HIV transcription. Now, in the nucleus, there's actually a punctate distribution of these complexes, and here's a rather remarkable effect, which is that proviruses, which in this experiment are labeled with EDU in a red color, become associated with little transcription factors in the nucleus with activated PTFB. And so you can see in this cell there were two proviruses, one of which remains silent, the other of which is actively transcribing, and the actively transcribing one is associated with the phosphorylated PTFB. I had to, since I knew Steve was going to be here, slip in a piece of clinical datum, which is that activation of PTFB can be treated as one of the earliest markers of T-cell activation. And this is data that came out of a romidepsin study. It's an HDAC inhibitor from John Mellors. And you can see in the bottom panel that the activation of PTFB followed the pharmacokinetics of the histone acetylation. There wasn't any HIV production induced by this regimen. That was very flat, but the cells were at least getting reproducibly and partially activated by this drug treatment. I couldn't leave without telling you something about epigenetics. I know that's Steve's favorite subject. And again, it's a mechanism used to silence HIV, to turn the promoter on and off. And what it really is about is modifying histones to create environments that are permissive or restrictive for transcription. And in the case of HIV, the major factors that restrict the chromatin in the resting cells are histone methyltransferases. And there are two major ones, EZH2, which modifies histones on lysine 27, EHMT2, which modifies histones on lysine 9. And so we know these are now doing chip assays on proviruses in these primary cell models. What you can see is that in the bottom right-hand panel, that histone methylation is high. That's the big black bar in the unactivated cells. When you activate cells through the T cell receptor, that modification gets reversed, and RNA polymerase concomitantly is being recruited to the promoter. Now, there are any number of histone methyltransferase inhibitors, and here we've looked at the impact of these methyltransferase inhibitors on HIV induction, and this is looking, we can do this in our model, but this has also been done in patient cells with a cellular RNA assay that we've developed, we call the EDITS assay. So what you can see is that all of these lysine methyltransferases will partially reactivate the cells, but they are particularly synergistic with other compounds. IL-15, which is one of our favorites because it tends to push PTFB production, and also in this case, verinostat, SAHA, which also has some impact on PTFB and probably is why it's been an effective compound. Now, the methyltransferase system can also be reversed with histone demethylases, and here's an example of the two demethylases, UTX1 and JMJ-D3, which selectively reverse the methylation of lysine 27, the EZH2 target. And so we asked the question, if you treat cells with these inhibitors of these demethylases, do you drive the viruses further into latency? In other words, do you make the chromatin environment more restrictive? And the answer is yes. Here's an example of treating patient cells with an inhibitor of these demethylases, GSK-J4, and you can see with the HIV reactivation assay that there's about 50 to 80% inhibition, depending on the patient, of reactivation in the cells, and concomitantly, there's an increase in DNA methylation. So there's a secondary epigenetic silencing that takes place when you eliminate the histone demethylation. Now, I want to close by talking about the ideal ways to reactivate HIV based on all of this. And the underlying ideas I've been explaining to you is that two things have to happen. You have to induce PTFB in the system in order to make the system permissive for HIV transcription, and then you also have to remove the epigenetic blocks. And so it's almost impossible for a single compound to hit the system at both stages, unless you're targeting a signaling pathway and in some way that's a bifurcated pathway. And so you need to look for combinations of compounds that can reactivate. And one of the things that we found is particularly useful is IL-15, which tends to prime the cells for reactivation and synergizes extremely well with other compounds. And so this is an example. It's just comparing data from the Q-cell assay, which is the ex vivo assay, and patient data, which is the EDITS assay. You can see that combinations of IL-15 and an HDAC inhibitor, SAHA, lead to reactivation levels that approach those that you can see with the T-cell receptor. Now, we actually have quite a bit of data now with these kinds of combinations of compounds and reactivation. You can see there's very good correlation between the ex vivo models and the data that you can get from the patient cells. And again, this remarkable kind of effect that if you prime the system with something like IL-15, which is initiating the PTFB program, then the epigenetic inhibitors really come into their own and permit very efficient latency reversal. So let me wrap up with a few general statements about latency. So the first, and I think the most important statement, is that latency is an integral feature of the HIV life cycle. You can even argue that the whole transactivation system is a device for the virus to reemerge from latency. And it's really kind of remarkable, but the necessity for NF-kappa-B and Nfat is entirely to get viruses to reactivate, to start reversing the epigenetic restrictions. If you delete those sites, the virus can replicate just fine in an activated cell. Non-suppressed patients have latent proviruses. There's nothing magic about ART. It's about 10% of infections in an ex vivo situation will lead to silent integrations or go into this program of latency. And as I've been arguing, the mere fact of infection of the cell tends to push it over into shutting down, and that enhances the frequency at which cells become latent. So if you have infections, you have latency. I think it's a little more controversial, but I believe that rebound is going on continuously. That when you have these latently infected cells, if they get into an inflammatory environment, permissive environment, the cells get reactivated, they start their program of emergence of the virus, and it sheds. Now, if you have ART, then that gets masked because there's no way for the infection to exponentially propagate and produce enough virus so we can pick it up with our very insensitive peripheral assays. So what's going on when you remove ART and you see rebound is simply that this natural underlying process is taking place, and there's a way of exponentially expanding the virus so it's detectable. In terms of eradication, I do believe in shock and kill, even though that's increasingly old-fashioned. So far, people have experimented with shock and no kill, and kill and no shock. And I don't think there's yet been a fair test of a well-defined shock and a well-defined kill. And I think that on the shock side, as I've been arguing, these synergistic compounds, which can be used clinically, things like IL-15, things like the HDAC inhibitors, things like the methyltransferase inhibitors, these are things that can be used to reverse latency efficiently. When designing these factors, I think it's also very important to make sure that they're compatible with effector cell functions. I don't particularly like protein kinase C inhibitors because they really wipe out CTLs and NK cells. And so, again, I think there's a balancing act between the effectors and the shock. And then I think Steve said it very well, that you want to have, in addition to this acute phase of removing reactivated cells, and I think NK cells are good effector cells to try for that, you do need to have long-term containment and likely that's going to be mediated by CTLs. And since there's nothing new under the sun, I note that actually in the clearance of LCMV infections, that's exactly what happens. The clearance is NK cells, and then there's a long-term maintenance with CTLs. So with that, I'd like to thank my colleagues who've worked on this project, and actually Kian and Frederick, who worked on the epigenetics and PTFB, are at this meeting, and I believe presenting posters, but I'm not sure of that. So thank you very much.